then we will start with the first speaker. It's my great pleasure to welcome Anika Liu from Cambridge, who's going to talk about her analysis of the DILI contest data set. I much look forward to your presentation. Here you go.
Thanks for the introduction, and hello, everyone. Yes, I'm a PhD student in the Bender Group at the Center for Molecular Informatics in Cambridge, and this work was done by a group of students in the group. And we've been working on this challenge with the L1000 signatures and also with the compound annotations from DILI rank. So our work consisted of two main parts. So first we tried to predict DILI either from chemical structure or from gene expression. And then in the second part, we tried to understand DILI better from our data, so by looking into chemical substructures and also into the biology. So for the predictive models, we trained binary classifiers, which ideally can predict for a new compound whether it causes DILI or not. And therefore, we used a set of compounds for which this is already known, and this was from the DILI rank database. To find the best models, we compared different training data sets, different methods, and different compound representations. And to find the one with the best performance, we performed cross-validation using a 5-fold stratified split. And also, we submitted predictions on the external validation data set with the relabeled compounds provided by the FDA. So we started with the structure-based models and compared different training data sets. The first one was a whole DILI rank data set. So here we were looking into the VDILI concern annotation and considering the most concern and less concern class as DILI, while we considered the no concern class as no DILI. So this gave us a slightly unbalanced data set with more DILI compounds than no DILI, and we balanced it in two different ways. So first we added more negatives from the CIDR database. So these are compounds for which side effects in the liver have not been reported. And we also looked at a smaller data set, removing the less concerned class from the DILI compounds, given that the evidence for DILI is weaker for these compounds. And to find out which of these training data sets would work best, we built predictive models, and we used ECFP4 to describe the chemical structures, and we used random forest and support vector machines as methods. So here you can see the performance of the different models. So the box plots show you the performance in a cross-validation. And so this is summarizing the performance across the 5 different folds. And the external data set performance is shown in the dots. And in the cross-validation, we found that the performance was quite similar across the different data sets. So the one without the less concern was slightly better in terms of balanced accuracy, but the whole DILI rank database was performing better in terms of precision and recall. But when we look at the external validations, we found that we've been overall overestimating our performance. And the best one was surprisingly the smallest data set, so without the less concern, using SVM. So this is why we used the smaller data set for our downstream analysis. And in the next step, we thought maybe we can find more interpretable compound descriptors, because ECFP4 only captures structural aspects. And we've been moving to molecular descriptors, also from the ModRec package. So these describe chemical and physical properties and could be potentially more interpretable. And also we've been looking into protein targets, which capture the bioactivity of the compounds. So given that not every compound has been tested for every protein, our descriptor matrix would have been very sparse. And in order to avoid this, we use PIGEN for protein target prediction. So this contains models which have been trained on bioactivity data from PubChem and Campbell, and we just give it a new compound structure, and it gives us the activity probability for over 1,600 human protein targets. So when running this tool, you can choose between different concentrations, and we chose to look at the activity at 10 micromolar, and also we didn't set an applicability domain in order to retrieve the full matrix. So here we found that the descriptors, which are shown in the different colors, again performed pretty similar. So the range was, in terms of balanced accuracy, was between 0.7 and 0.74. However, on the external data set, ECFP and SVM remained the best, so we were not able to improve the predictions. So we then thought maybe gene expression will work better. So we looked into the L1000 landmark genes here, and we built models on all the conditions which covered all the compounds we had. So these were 14 cell line time dose combinations in the LINCS database. And we then built models using, again, the most concern and no concern class and random forest and XGBoost as methods. However, to keep this short, we just found that the performance wasn't really good. So the best performance in terms of cross-validation was 0.66. And when we submitted this for external validation, we found that the balanced accuracy was around 0.5, so pretty much random. And because of this, we didn't submit further predictions, and we didn't try to optimize our models further, and focused instead on looking whether we can learn more about DILI from the data. So here we've been looking into the chemistry. Maybe there are indicators which can be helpful in early drug development and lead optimization, and also into important genes and proteins which might help understand DILI. So substructures were derived from our dataset from DILI-rank using two different tools. So this was MOS and SARPy, and we also included 27 substructures from literature. So we found that there were some substructures which are very common among DILI compounds, like sulfoxide and benzene, but what we're really looking for is something with a high positive predictive value, so that if you find a substructure, it's very likely that it causes DILI. And here we found that there were 3 substructures in the literature dataset for which this was true, and also there were these 4 substructures from SARPy, which had a relatively high positive predictive value and were significantly enriched in the DILI compounds. We then moved on to the biological interpretation. So here we've been starting to look into the L1000 landmark genes, and we found that across the 14 conditions that we had defined before, that there were 2 genes which were significantly upregulated in DILI across the majority of the conditions. And the first one was KAP1, which is known to be involved in the hepatotoxicity of acetaminophen and is related to the NRF2 cell defense system and oxidative stress. And the second one was XBBP1, for which absence has been shown to be hepatoprotective in mice, and this is related to ER stress. However, the interpretation we couldn't take much further, given that only 2 out of the 1,000 genes were significantly enriched. And this is why we went back to our chemical structure-based models, and we looked at the predicted protein targets and their importance in the models. So here we focused on the ones which were significantly enriched in DILI, so the ones shown in red. And among those, we then looked at the ones with high feature importance, so the ones on top of the plot. And this gave us a ranked list, and the top of the list you can see in the table over here. So we haven't worked with DILI before, so this list didn't tell us much. And because of this, we first looked into what we would expect from the biology. So here is a very general overview of the processes involved in drug-induced hepatic apoptosis. So there's one way which is immune-mediated, and then there's the one I'm going to focus on is the intracellular signaling. So here, a drug or one of its metabolites can interfere with the signaling through different receptors, activate cellular stress, or lead to mitochondrial inhibition. And this then leads to intracellular activation of caspases, the J&K pathway, or mitochondrial signaling. And eventually, all of these processes can cause hepatic apoptosis. In our data set, just looking through the top 40 proteins, we found that there were 3 that mapped onto these processes. So this was aldose and quinone reductase, which are involved in cellular and mitochondrial stress. And then there was also MAP kinase 9, which is part of the J&K pathway. In addition to those, we found 2 CYP enzymes, which are involved in xenobiotic metabolism and are known to have high expression in the liver. So these could potentially contribute to the production of toxic metabolites. But of course, these are only 5 out of the top 40 proteins, and we wanted to know whether there are generally patterns in our data. And this is why we performed an over-enrichment analysis using the reactome pathway maps. And we used different feature importance cutoffs for this and found that there is a set of pathways which is significantly enriched across different cutoffs. And yeah, you can see these pathways here. They are ranked by significance. So the most significant one was mitochondrial biogenesis, and the one with the highest gene ratio and gene count was metabolism of lipids. So we were very happy to see these processes coming up as most significant, given that these cover essential parts of hepatic apoptosis. So mitochondrial biogenesis and remodeling is activated by mitochondrial stress and energy depletion, and is also known to be involved in DILI. And also TP53 is very known in DILI and integrates different cell stress signals integrates different stress signals, has been linked to the JNK pathway before and also to mitochondrial signaling. What was half expected was the signaling to ERGs, given that the JNK pathway and the P38 pathway, which are parallel MAP kinase cascades, are known to be involved. But we found that we couldn't have detected those given there was no gene set. And also crosstalk between the pathways is very well known. And because of this, we then decided to take a closer look. And so here you can see which protein targets were responsible for the overrepresentation of the different processes. And having a closer look at signaling to ERGs. In this one, for this, it was the MAP kinases 11 to 14 that caused the over-enrichment, and then these are in fact the 4 out of 4 P38 kinases, so that we can say that in this case, P38 signaling was the cause for ERG signaling enrichment. So the other processes didn't map to hepatic apoptosis directly, but they are still linked to DILI. So fatty acid metabolism and metabolism of lipids are clearly very general processes, so they could take in very different roles, but they have also been explicitly reported to be involved in the hepatotoxicity of valproic acid. And what was a bit more specific is that we found arachidonic acid metabolism and the synthesis of prostaglandins and thromboxanes. So this is the mechanism of action and the mechanism of DILI for non-steroidal anti-inflammatory drugs. So these interfere with the formation of prostaglandins and thromboxanes from arachidonic acid. And the interesting part is that these are the second most common cause for DILI worldwide. So we were able to find general processes involved in hepatic apoptosis, as well as this particular mechanism of action for an important drug class. And also I want to add here that the most common cause for DILI are antimicrobial drugs, so this wouldn't have been possible to get from human protein targets. So to summarize, first we tried to predict DILI and found that chemical structure resulted in slightly better performance than gene expression. And however, this is probably the cause that we had way more labels to model chemical structure than gene expression. And overall, our predictive power was still low, which shows us that we are not able to capture the complexity of DILI. And because we were thinking that our predictions are not really good, we thought maybe we can learn a bit more about DILI still from the data set. So here we found that we were able to identify 4 new substructures, which show a high positive predictive value for DILI. And so these could be useful indicators for hit screening and lead optimization. However, the coverage of DILI compound is low, so if you don't find it, it doesn't help you. But what was the most interesting part for us is that we were able to understand the biology better from chemical structure. So this was done using target prediction models and pathway maps. And here we identified both key proteins and key processes. So although I have touched this before, I wanted to mention the limitations of our models and our work. So an important aspect to consider is that DILI is very complex. So we are only looking at chemical structure and gene expression in cell culture here, but DILI is dependent on the dose the patient's taking, as well as other confounding factors such as age, disease state, or other drugs the patient might be taking. So this is overlooked in our models, but it's also unclear how it goes into the drug labels and eventually also the binary labels that we are trying to predict. And the second point is that the chemical applicability domain and the biological applicability domain is limited, so that it's unclear whether we can extrapolate predictions to novel chemical space and novel modes of toxicity, which is, of course, interesting for drug development. So this work was done again by a subgroup of the Bender group. So I want to thank them and also a few other people that have given input. And also I want to thank our funders for supporting and sponsoring us, and CAMDA for supporting me with a travel fellowship. And if you have more questions afterwards, you can talk to us at poster Q05, and thank you for your attention.
Yeah, there's a bunch here. I don't know which one's making it. Oh, it's covered.
Does this work? No? Okay, I will borrow your microphone for a second. So thank you very much for this talk. Sorry for stealing your microphone for a minute. Very, very nice to see how you've sort of turned this into a discovery mission instead. Are there any questions from the audience, please?
Just like you, even the other speaker chose the dose to be 10 micromolar. Is there any reason, chemical reason for choosing that dosage?
It's kind of arbitrary. So for the links, we chose 10 micromolar because this was the concentration which had all the data for all the compounds.
Okay.
So that was predefined. And for the protein targets, we could have chosen like 1 micromolar, 10, or 100. And 10 is a bit more than the Cmax, so the dose you would expect to see in the body. But the toxic effects usually, like, they might appear at a slightly higher dose. So this was why we chose 10 micromolar.
Thank you.
Any more questions? Yes.
Very interesting work, thank you. So I have two questions. One is, do you consider prodrugs in your data set?
So we didn't consider metabolism at all here. So it's only like for the parent drug that we do the target prediction.
Another thing is actually, with regard to your comments about the limitation part, I think they are very valid points. Is there any thinking how to address these problems?
So we found that like chemical structure, gene expression, it helps a bit, but in order to understand DILI better, we would need patient data basically, because there's all these genetic variances and so on. But then again, if you go to the patient level, it's not really helpful for drug development anymore, because then they need to do the clinical trials anyway to get to that data. Yeah, so I think we could include more on the metabolism, as you mentioned. So we've been looking into like concentrations in the human body, but yeah, so potential ways to extend it, but I'm not sure what would be useful for drug development in the end.
Thank you. Any more questions? And thank you very much again for your contribution.